afternoon, colleagues, students, ladies and gentlemen. Welcome everyone to our webinar today. On behalf of CRCD Utah, I am very pleased to introduce to you our speaker for today, Prof. Dr. Yeo Chiu Cheng, who is going to talk about the potential and limitations of whole genome sequencing, or in short is WGS, of bacterial pathogens. This is a subject in which we should all be deeply interested due to the advancement of high throughput DNA sequencing technologies, which already happens about 15 years ago. So why do we care? The advancement of DNA sequencing has revolutionized biology in ways that were unimaginable at the turn of the millennium. Whole genome sequencing is poised to be the dominant technology in routine diagnostic microbiology laboratories. However, whole genome sequencing has yet to be widely adopted in clinical microbiology, particularly in middle-income countries such as Malaysia, despite its tremendous potential. Prof. Yeo will present some of his whole genome sequencing results of Acinetobacter baumannii, isolates that were obtained from the main tertiary hospital in Terengganu, Malaysia since the year 2011. So it is a gram-negative nosocomial pathogen, which is originated in a hospital. This pathogen is of concern due to its unique ability to acquire and develop resistance to all classes of antimicrobials, thereby severely limiting therapeutic options. He will also discuss about the current limitations of whole genome sequencing, such as its existing cost, the requirement of decent computational infrastructure, along with the training of laboratory and technical staff in bioinformatics. Let me give a brief introduction about Prof. Yeo's background. He has been specializing in microbiology for years. He obtained his PhD in microbiology from the National University of Singapore, NUS, in 1998. He was employed as a postdoctoral fellow at the Department of Microbiology, NUS, until the year 2003, when he joined the Malaysia University of Science and Technology, MUST, as an assistant professor in the Faculty of Biotechnology. I was really fortunate to have him as my master supervisor during my postgraduate study in MUST. And if I'm not wrong, I was his first postgraduate student, right, Prof?
Correct.
I'm very proud to say that. So after that, he moved back to his hometown of Kuala Terengganu since 2009 and joined the then newly established University Sultan Zainal Abidin, or in short, UNISAS, as an associate professor in the Faculty of Agriculture and Biotechnology. He is now currently a professor in microbiology at the Faculty of Medicine, UNISAS. So his current research interests are in the genomics of multi-drug resistant bacteria, pathogens, in particular Acinetobacter baumannii and Staphylococcus aureus, and the molecular biology of bacterial toxin antitoxin systems. He served in the editorial board of several journals such as Frontiers in Microbiology and Frontiers in Molecular Biosciences. Also, Prof. Yeo is a member of the American Society for Microbiology and the Microbiology Society United Kingdom. So without further ado, ladies and gentlemen, please join me in welcoming Prof. Yeo to share his insight with us today. If you have any inquiries or comments about the contents of the talk later, please post it in the chat box or wait for the question and answer session after the talk. We will respond to that later. So Prof. Yeo, I will pass the stage to you now. Thank you.
Okay, Siu-Hun, thank you so much. She's been just now before this thing went online, we were chatting, and well, this talk has been more than a year coming, because I was supposed to go to Utah in Kampa to present this talk to her, to you all, but due to unforeseen circumstances, it was postponed to February of this year. And then, of course, COVID had happened. So everything got postponed until now. So anyway, my thanks to Siu Hoon and everybody in Utah for inviting me to give this webinar on whole genome sequencing of bacterial pathogens. It's a topic that's close to what is dear to me, because that's what I've been doing in UNIZA for the past couple of years. Okay, so hope everybody can see the slide okay. Yeah. Oops. Right. So this is just a figure that shows you milestones in genome sequencing and assembly. So starting all the way since 1965 until now, 2020. So how much we have progressed from the first DNA sequence rather of a 76 base pairs partial transfer RNA of yeast or E. coli 5S ribosomal RNA. There's only 120 base pairs. That's like 40 over years ago until where we are now. Okay, so one thing I'm going to highlight, because the focus of my talk today will just be on bacterial pathogens. So bacteria is going to be my highlight. So that's why you see the red box here on H. influenza. So basically that was in 1995, in which the first complete genome of a free living microorganism. This was published in Science in 1995, right? Haemophilus influenza. And this was sequenced using the Sanger dideoxy method. So this is what we could now call as first generation sequencing technology. It's expensive, it's cumbersome, it's time consuming. So for instance, for H. influenza, they needed 28,643 sequencing reactions for Sanger. They use it on shotgun libraries. On average, 14 Sanger machines, ABI 373 DNA sequences. Those who are old enough to remember the ABI 373 sequences way back in the 90s and the early millennium is what we've been using. So they use an average of 14 per day over a 3-month period. And back then, sequence assembly was still in its infancy, but they managed to get it done and assemble the genome of Haemophilus influenza into 140 contigs. Then this was actually quite a feat already. So that's like how many years ago? 1995 was 25 years ago. But we have gone a really, really long way since then. So there have been at least 2 waves of sequencing technology innovation since the turn of the millennium. So following the first in the mid-2000s, you have what they call next generation sequencing technologies, or the second generation ones. These are called NGS for short, right? So here, sequencing is carried out in a very massively parallel manner, and this sharply reduced the cost to generate your sequence data. So this massively parallel sequencing was actually pioneered by 454, which was then bought over by Roche, and then after that came Illumina, Ion Torrent by Pacific Bio, not Pacific Bioscience, it's something else, I can't remember, Ion Torrent, and then Applied Biosystems, they have the Solid. Then, about 5 years later, 2010 onwards, you have the third generation sequencing. So here you have what they call real-time single molecule sequencing. This was pioneered by Pacific Biosciences, the PacBio. You have the RS2 sequel, and now you have the most recent one is sequel 2. And the other company that actually pioneered this is called the Oxford Nanopore Technologies, or ONT. This is called Nanopore sequencing. Here, you have things like Minion, Gridion, and Promethean. So this third generation sequencing is what we are in now. Okay, so I'm going to cover a little bit of that in a bit. Now, but in late 2018, Illumina actually agreed to buy over PacBio for quite a hefty sum of money, to 1.2 billion. But by earlier this year, in January 2020, this deal was abandoned. I suppose Illumina thought that PacBio is not what do you call it? It doesn't make enough, won't make enough money, because the nanopore sequencing is really looks like it's going to be the winner for the third generation sequencing platform. Okay. So a rough idea of what the difference is between second generation and third generation. Now for Illumina platform, what you get from raw data is just short sequence reads, which is less than 300 base pairs. Normally we do what we call pad and reads of 150 base pairs, right? But one thing good about Illumina is that you get a very low per base error rate, which is typically less than 0.1%. So you always get deep shotgun coverage, and assembly typically results in multiple contigs and collapsed repeat regions. So that's the drawback of Illumina, or the short sequencing reads, is that when you have repeat regions, they collapse together and then what you get are multiple contigs. Okay. It is still currently the dominant player in the NGS market, but well, I'm not sure for how long this will be. And I'm sure you heard of, if you talk about Illumina, I'm sure you heard of the MiSeq or NovaSeq platforms. Third generation sequencers, they're characterized by long sequence reads. I mean, Illumina, you're talking about 300 base pairs, but for third generation sequencers, you're talking about anything between 5 to 100 kilo base pairs, okay? And of course, unfortunately, as compared to second generation sequencers, you get a much higher per base error rates, which is about 5 to 15% higher than Illumina. So this is actually compensated by achieving higher read depths, and recent studies, and you get what they call continuous improvement in the accuracy of long reads, as well as error correction tools available. So one good thing about using third generation sequencing is that your assemblies will have much fewer gaps and span all those lengthy repeat regions. They don't have as much problem in handling repeat regions as compared to the second generation sequencers. So let's have a quick look at some of these third generation sequencing technologies. First off, we have the PacBio system, okay, or the SMRT system. It was first released in 2011, so it's coming to 10 years already, the PacBio. So the average read length was 1.5 kb. The RS2, which released in 2013, increased the read length to 20 kb. And then what you have now is a sequel to, and the read length is more than 50 kb. So you got a very long read length, so which makes assembly easier. Of course, the drawbacks for the PacBio system is you have low run, the throughput is low run, is a very high per sample cost. Instrument cost is also high, and instrument footprint is large. So these machines are huge. Okay, so it is PacBio is definitely more suitable for core genomics facilities, which aims to generate high quality complete genomes. Now, compared to the MinION, the Oxford Nanopore sequencing. Okay, this is the size of a MinION sequencer. If any, I don't know, afterwards during the Q&A, I'm really very, very interested to find out how many of you guys in Utah are using the MinION sequencers. I aim to start doing it next year if I can get grants for it. Okay.
And next NGS as well.
Sorry?
We don't have NGS.
No one.
Third generation.
No one. You guys should really look into going to Oxford Nanopore because it's cheaper, it's small, the footprint is small. Okay. I think this is the MinION is actually, the nanopore sequencing is actually the wave of the future. So I'm not surprised that Illumina bail out from buying over PacBio. So the thing about nanopore sequencing is it provides very long read lengths, from 500 base pairs. The record is 2.3 mega base pairs, a single read of 2.3 mega base pairs. Okay, but usually you have about 10 to 30 kb libraries. So the problem with nanopore sequencing is, although it gives you a very long read length, you have a high error rate. Error rate is much higher compared to PacBio or Illumina. But this is actually compensated, okay, and is vastly improving with dramatic increases in the accuracy of base calling and also of software that have been developed to actually try and decrease the error rate of the nanopore sequencing. So especially within the past 2 years, there's been like an explosion of software tools that are available and is catered for the nanopore sequencing. And each flow cell, each MinION flow cell here, the one that plugs into the USB, can accommodate concurrent samples and generates between 4 to 8 gigabase of data. Okay, and if you look at it, it's great for field diagnostics application. And although my talk is basically talking about bacteria, I need to show this because this one is actually give you an example of how useful the MinION sequencers work in an epidemic situation. So it was successfully used to sequence the Ebola virus outbreak in West Africa about 5 years ago, and they do it directly in the field. Results were available less than 24 hours after receiving clinical samples, and the sequencing process takes as little as between 15 to 60 minutes. So you can see there this is the actual picture from the Nature paper, right? And this is how they have in the lab. You have one bench for all the PCR machines, the PCR bench, and the next bench next to it are just 3 MinION sequencers. It's wonderful. It's really wonderful. It allows researchers to track the Ebola virus transmissions of separate lineages across countries by monitoring the mutation rates. So this is a real prime example of how this type of technologies has really improved in just the past 20 years. Now you can sequence things with just a laptop and a USB connection. So Oxford Nanopore actually offers a range of sequencing devices now. They first started out with just the MinION. Okay, I think they released this. They released this around the time of Despicable Me, so that's why they named it MinION. It was a really good marketing on their behalf of calling MinION, and the ion actually comes from the way they detect the sequences because they use nanopores and they detect it by the flow cell actually detects what do you call it, electrical currents from between different GATCs. So they call it MinION. And and they got a smaller version out now called the Flongle. This one is a souped up or much better version of the MinION called MinION Mark 1C. And these days, they just came out this, I think, last year or 2 years ago, which enables high throughput benchtop sequencing. So the Gridiron and the Prometheon. This one allows you to, like, I think, if I'm not mistaken, about 96 flow cells at a time. So it allows you for high throughput sequencing, all at the benchtop. So as I said, I think nanopore sequencing is the wave of the future. So these days, if you want to do whole genome sequencing, like what I do is I prepare genomic DNA, send it to a commercial provider, they send it to probably China or Singapore, and you wait 3 months if you're lucky and get back the raw sequence data. Now, if you have the MinION sequencer, I just force my students to do the really high quality DNA, do the library prep, run it on the MinION, and by next day, I can get the raw sequence data. Okay, so that's a wonderful thing about how genome sequencing technologies have progressed within the past few years. So this is actually a really good table of summary of all the commonly used platforms for next generation sequencing. So it starts from the Roche 454, Ion Torrent, Illumina, the PacBio, and then the Oxford Nanopore. Okay. So going back to this milestones that I showed earlier on, so you can see where all the various sequencing platforms were used. So all those in yellow are the Sanger-based shotgun sequencing. This was predominant in the mid-90s, from the 80s, sorry, all the way to the middle of 2000, around 2005. That's when the first next generation sequencers came in. And then these became predominant until about 2015, sorry, when third generation sequencers came into the picture. Okay, right. Okay, so that gives you that gave you a sort of a background of the different platforms for sequencing. So now what I'm going to do is I'm going to talk a bit on how this have impacted or is being used in clinical microbiology. So the early applications of whole genome sequencing in clinical microbiology and public health uh, is basically to elucidate and interrupt transmission pathways whenever we have nosocomial outbreaks of pathogens such as Acinetobacter, Staph aureus, or Klebsiella pneumonia. So all these were early papers that looked at how whole genome sequencing can help in clinical microbiology and public health. And in these instances, the analysis for the whole genome sequences always were conducted from isolates that could be cultured. So you need to culture them in the lab. So once you have a pure culture, then you extract the genomic DNA and then you run the whole genome sequences. And it does provide a huge amount of information regarding the species, the type of resistance, what type of virulence, and all those different type of information for outbreak and case management. So basically, sequencing, whole genome sequencing is now moved to become one of the frontline diagnostic tools in the clinical microbiology. Now, of course, these days we've moved on from doing sequencing of pure cultured organisms and looking at metagenome sequencing, right? And this one, you're talking about applying, you can sequencing, or rather you can sequence direct from either DNA or RNA that's extracted from patient samples, from environmental samples. So this one will circumvent the need to culture the pathogen. So this is what you call a shotgun metagenome sequencing. And this is really wonderful for slow-growing and fastidious bacterium such as Mycobacterium tuberculosis, right? So these are prime targets for metagenome sequence application, which can use to predict the resistance phenotypes, determine the clonal characteristics and the virulence, etc. So basically for metagenome sequencing, you get the clinical samples, extract it directly, and then do the shotgun sequencing and assembly, right? Or you can do the long read sequences and get a much larger, sorry, you can get a longer read compared to short reads, right? However, when you talk about metagenome sequencing in clinical microbiology, there are still some drawbacks to this approach. Of course, first and foremost is complexity, cost and complexity of the process. So there's a requirement for us to do optimization and standardization for each step of not only the sample prep, but also the data analysis. So this is still quite a drawback. It needs to be the hurdle that needs to be overcome. And another hurdle you need to overcome is sometimes things like Mycobacterium tuberculosis, for instance, you will have a low ratio of pathogen to host nucleic acids, right? And most often you get the presence of commensal bacteria as well as minutely contaminated laboratory regions. So this can actually lead to incorrect results. So these are some of the hurdles that one needs to overcome, but I'm confident that these can be overcome within the next couple of years. So this would definitely be a routine in the future. Maybe within 3 to 5 years, you're going to see just take a clinical sample, any clinical specimen, good, pass, whatever it is. And instead of doing your traditional culture method, extract the DNA, write it for me, and just do metagenome sequencing. Okay? Right. So for the next part of my talk, what I'm going to give you is a rough idea of what we have carried out in UNISA here in Terengganu in terms of the whole genome sequencing of Acinetobacter baumannii. So this was a project that I started way back about 2011, so almost 10 years ago. So I've been collecting strains of A. baumannii from the hospital, the main general hospital in Terengganu. Now, Acinetobacter baumannii is one of those notorious pathogens. It's actually listed. It was actually listed by the WHO as one of the top priority pathogens in 2018 because, and it's this notoriety came about because it's able to acquire and develop resistance towards all classes of antimicrobials. Okay, all classes of, so including the drug of choice is for carbapenems, and the drug of last resort are the polymyxins. Now we initiated this project because there's very little data on A. baumannii from Malaysia. So based on a few publications, so we only found that, okay, majority belong to this clone called the GC2 clone, and the predominant carbapenem resistant gene is OXA23. So what we have done is, of course, I do have a couple of hundreds of strains, but I'm limited by budget. So one can only sequence so much. So we, in our first phase, we sequenced about 50 isolates that's obtained from 2011 to 2016. And we look at the distribution of sequence types. And we found that 2011-2012, we have ST195 as a predominant sequence type, right? And most of these are multi-drug resistance, okay? With nearly 25% were resistant polymyxins, meaning the antibiotic of last resort. But strange thing happened through the years. From 2015 onwards, the ST195 clone is basically not there anymore. It's been replaced by a wide variety of other clones, right? So why this clonal population shift occurs is currently not really sure. We haven't gotten around to sit around and sequence and analyze all the 50 isolates data. This is a very slow, laborious process, something I'm going to talk about later. Right, you can get lots and lots of data, but to analyze them will take manpower and time. Okay, so if you look at the phylogenetic analysis of the clones, as you can see, this whole lot here, you can see the bits in purple here, they are all very, very closely related. So all the ST195 clones in 2011 and 2012 were inferring that they have, what we have is actually an endemic spread of A. baumannii in the hospital for those 2 years. But following that, all the clones are very diverse, right? They are very, very diverse. So we're not sure why this thing happened. We are just in the process of sequencing from 2017 onwards the strains, and we'll see how the trend goes, how the trend develops from there on. But we focus basically for the past year, we've been focusing on just 2 isolates. 2 isolates, and these 2 isolates are interesting because they carry the what they call the New Delhi metallo-beta-lactamase 1, NDM1, and they also cover the OXA58 carbapenemases. So NDM1 is notorious in the sense that it makes the host bacteria to become resistant to all beta-lactams, right? And they are not inhibited by beta-lactamase inhibitors such as clavulanic acid, your sulbactam, tazobactam. They are not inhibited by that. So you cannot use combination of beta-lactamase inhibitors and beta-lactams. It doesn't work for strains that contain NDM1. Okay, so both now we have 2 strains actually that contains NDM1 positive from our collection of 50. One is we identify as Acinetobacter baumannii, we call it AC1633. The other is actually a close relative of Acinetobacter baumannii called Acinetobacter nosocomialis. The strain we label as AC1530. So background 1633 is actually isolated from blood of a 60-year-old female patient in neurology in April 2016. The patient succumbed to septicemia 41 days after hospital admission. AC1530 isolated from the blood of a 14-year-old male patient who developed hospital-acquired pneumonia after admission for polytrauma from a motor vehicle accident. Now, patient was recovered and was discharged after 60 days of admission. So this is the background for the two, the acinetopulmonale isolate, the AC1633, and the nosocomialis isolate, which is AC1530. So we did detailed whole genome sequencing of these two isolates. Currently, the manuscript is undergoing revision, but we have published it in bioRxiv, the full paper. If anyone is interested, we put it up on the preprint server bioRxiv. Now, because Illumina sequencing showed that the two carbapenem resistant genes are possibly located on either one or two very large plasmids, but difficult to determine because there are like more than 20 contigs. So is it one or is it two? We don't know. So what we did is we bite the bullet and spend money to do PacBio sequencing. Now, Illumina sequencing cost us around 600 plus ringgit per isolate. PacBio is 10 times more, 10 times that. So PacBio costs us about 6,000 ringgit per isolate. So we spend about 12 to 14. Here in this case, we spend about 14,000 ringgit to do PacBio sequencing for two strains. Okay, so this one we didn't do assembly of just a PacBio. What we did is we did hybrid assembly, meaning we use both PacBio and Illumina sequencing to carry out the sequence assembly. And this is the result of it. So A pulmonale, AC1633, one chromosome of about 4.4 megabase pairs, and it has 4 plasmids. This is a very big plasmid, about 174 kb. A nosocomialis 1530 chromosome is about 3.9 megabase pairs. It has also one plasmid, the large one. It's about 174 kb. Okay. Now, when we do analysis of the phenotype, the resistance phenotype, and look back at the genome data for the genotype, we have a complete match of all the resistant genes as well as all the resistant phenotypes. Okay. So basically, look at carbapenems. It's resistant to imipenem, meropenem, doropenem, and it has what is highlighted in blue is the plasmid. This is the NDM1 and OXA58 carbapenemase. Likewise for nosocomialis. So as I show in the slide here, bulk of the acquired resistant genes came from this large plasmid, this large 170 kb plasmid. So cephalosporin is actually chromosomally encoded in both pulmonale and nosocomialis. Aminoglycosides is the large plasmid. Tetracycline is resistant for pulmonale and is on the second plasmid. Is susceptible for both tetracycline and doxycycline for nosocomialis. Sulfonamide also in the large plasmid and macrolides in the large plasmid. Okay. So when we look at the detailed sequence of these two plasmids, we found that these two plasmids are almost identical, okay, except for the insertion of and deletion of mobile genetic elements. So if you look at the genetic map of these two large plasmids, so one PAC1530 is from nosocomialis, PAC1633-1 is from pulmonale, okay? So you can look at it, they are almost identical. So the difference here is like, okay, you got an insertion element ISABA11 here. This is an IS element here, IS element here. So you just have insertions and deletions of IS elements, right? So antimicrobial resistant genes all in yellow. Beta-lactamase, where's your, this is your NDM1 here, and your OXA58 over there, okay? So this plasmid is basically a multi-drug resistant plasmid. It also contains genes for conjugative transfer, which makes it transmissible by conjugation, right? And if you look at the, NDM1 gene, the environment is basically in a transposon, we call it a DN125, and this is about 10 kb composite transposon, and it's also found in many Acinetobacter species. So this is nothing really novel about it, right? Because this transposon has been documented in many Acinetobacter species as the vehicle for the transmission of the NDM1 gene, right? Here, it gets a bit complicated. I don't think I need to show, I won't go further in because otherwise I can spend the next half an hour by talking about this. But the other resistant genes, what I'm saying here is that other resistant genes, aminoglycoside resistance, sulfonamide resistance, they are also in mobile genetic elements. So all these are found within mobile genetic elements inside this large plasmid. Okay. Now, the interesting part, the novel part about this is that we sequenced two separate species of Acinetobacter from Terengganu. Okay, but when we did the blast to compare, see what's the nearest sequence that you have in a database, we found that we actually hit very high sequence similarity with two plasmids from Acinetobacter pti that was isolated in Perak in 2011, where you guys are. Where you guys are in Utah. So this was actually published in about 2015, 2016. It is Prof. Chan Kok Gan's group in University Malaya. So I'm not sure which part of Perak they got it from, but we found almost perfect hits with this. And in this Acinetobacter pti, NDM1 and OXA58 were found in two separate plasmids. They are two separate plasmids. Okay, so what we hypothesized happened inside our Terengganu Acinetobacter strains. So these two are the two plasmids that in the Acinetobacter pti from Perak. Okay, so what we hypothesized is that the OXA58 plasmid, there are two IS elements here called IS1006, and there is also an IS1006 in the NDM1 plasmid. This is all in a single Acinetobacter pti. Okay, so there is actually a transposition or recombination that happened at IS1006, leading to become one big plasmid. Okay, so because the sequence identity is really giving all hints of this thing happening. Okay, this is what happened. But how it got from Perak in Acinetobacter pti to Terengganu in a different Acinetobacter, that is a big mystery, it's a big question mark. But this is what we hypothesize to have happened, right? Based on the sequence data that we have, okay? So this is of course suggestive of transfer, because we get the same hospital, two different Acinetobacter species, isolated 2 different years, is suggestive of the transmission of plasmids. And our sequence analysis also show the presence of a lot of conjugative transfer related genes. But in our lab, when we tried several times, all different types of conjugation assays to try and show experimentally in the lab that conjugative transfer does happen. We were not able to do so. So this is one thing that eludes us for now. The laboratory demonstration that conjugative transfer has taken place. But all evidence based on sequence are showing that transfer has occurred. Okay, just that now we can't prove it in the lab. There are 3 other plasmids in Acinetobacter baumannii. Besides the large plasmid, the tetracycline resistance genes are found in the smaller plasmid, right? Now, these resistant genes are found also in a mobile element. They call it a diff module, pdiff. Okay, So you find this in many other plasmids in various Acinetobacter. Okay, we also found it in our collection of 50 Acinetobacters. We do have several tetracycline resistant Acinetobacter that contains these genes in this particular module, this really discrete module. Okay. The other two plasmids, 16333, 16334, are, however, cryptic. We're not sure what they are. But the interesting things about all these plasmids is it also encodes a what they call MOP-AL genes, meaning they are mobilizable, which means that if in the presence of a conjugative plasmid, this plasmid can also be transferred. Okay, so this actually goes to show that in Acinetobacter, as well as in most gram-negative bacteria, the genome is very fluid. So horizontal gene transfer occurs very readily. I think the exception is maybe like Mycobacterium tuberculosis. I don't think horizontal gene transfer occurs often, right, Sihun? Right.
Yes, Prof.
Yeah. Okay. So currently, this is what we have. So we did, as I said, we did what we have done for Acinetobacter is we sequenced 50. We found 2 that's interesting. So we really throw in money and focus our data analysis on just these 2. And these are our findings, and hopefully it'll be published soon. Just have some minor revisions to go. We actually submitted to antimicrobial agents and chemotherapy, but they transferred our paper to MSphere because, you know why? Because MSphere is open access, so they want us to pay money. These days, unfortunately, for publication, that's how it is. Yeah, there are very few free journals out there. Okay, last part. I'm down to my last 5 minutes. So it's like if you have a whole genome sequencing in a clinical or public health laboratory, this is your typical workflow. The specimens need to be sequenced, pure culture. This is the old-fashioned way. We're not talking about metagenome sequencing yet. DNA extraction, check for quality control, and then run it on. This is an Illumina machine, right? Now, this is the part that's, this part is actually quite straightforward. This is the part that's most laborious, your bioinformatics analysis, right? So I always like to show, I've been showing this picture of this baby in a hose of water for 10 years already, ever since next generation sequencing came into play. And the biggest challenge, I said, in introducing whole genome sequencing in clinical microbiology is data analysis, right? So a majority of the bioinformatics software is in Python, in Perl, C++, so R. So if you don't know how to use your command lines, a bit tricky, right? There's very few Windows-based graphical user interface software out there for data analysis. So I force my postgraduate students to at least learn rudimentary command lines in Linux. At least they have to know that actually. Otherwise, you know, how you can't even do assembly. So sequencing costs may be decreasing, but of course, you do require high-end PC or workstation to handle all this data, right? So this is one of the main drawbacks, the limitations of it, right? Bioinformatics. So important to have dedicated, well-trained staff to ensure successful implementation of whole genome sequencing in routine clinical microbiology, because there are 2 main steps in the whole genome sequencing, the wet lab part as well as the dry lab part. So both are equally important. You cannot say that, oh, wet lab is much more important. No, you can just, wet lab is just up to the point of getting genomic DNA extraction. It's a dry lab that takes a long time. And of course, those dry lab bioinformaticians will say, oh, we're the most important. No, if you screw up your genomic DNA isolation, then you get junk. You won't get good quality raw DNA, raw sequence data for you to analyze. So both are equally important. Okay, final 2 minutes, simple cost analysis. I want to do a comparison of cost analysis. This slide I made last year. I think Sihun or those who attended the, because I gave a talk at Sungai Long last year. So this is actually the same. I've just upgraded it a little bit. So approximate minimum cost for Illumina sequencing at that point is $80 per microbial genome. This one, of course, you're talking about based on 1 week turnaround time, batching many samples together. And the conclusion in 2019 is it's still too high cost for routine use in clinical laboratories. But in October of this year, Smith and others came out with this really interesting paper where they assessed the feasibility of using MinION for Mycobacterium tuberculosis in a clinical lab. Okay, so they compared the MinION with the MySeq platform. They assessed 431 samples over a 32-week period. And in terms of genotypic analysis, resistance predictions, the MinION is comparable with MySeq. Okay, there's not much of a difference in terms of its performance. There's interesting part is when you look at the cost. Okay, this is the cost minus labor cost, of course. They estimate that MinION without flow cell reuse is cost you about $63 per sample. If you use reuse the flow cell is 57, it's cheaper. But compare it with Illumina. Illumina is $130 per sample. So that's why I said in the early part of my talk that nanopore sequencing is the future for whole genome sequencing. I really aim to try and fiddle with that next year if I can get money. Okay. So there are many issues for whole genome sequencing. I won't bother to go into details behind it. But one of the issues they always like to point out is the correlation between genotype and phenotype, especially for antimicrobial resistance. It's still under debate. But in our case, when I look at Acinetobacter, we got complete 100% concordance between the genotype and the phenotype for this. But they do have problems, especially like in example in Neisseria, azithromycin resistance is due to mutations in the 23S ribosomal RNA, which exists in multiple copies. So there may be a problem, especially when you do assembly algorithms, where you have multiple copies of this. This can be collapsed into single consensus sequence, so you won't really detect the mutations well. But with third generation sequencers such as MinION, this shouldn't be a problem because they are long reads. So even you have multiple copies, they can read past it and give you the exact data. Okay, so outlook, sequencing technology, everything, bioinformatics, informatics are all rapidly evolving. If I were to give this talk next year, there'll be even more improvements to it. Okay, so the pace of the development of whole genome sequencing has just been really, really tremendous, especially over the past 1 or 2 years. I think the focus now, I'm looking at the development over the past year, the focus is basically nanopore sequencing. They've come up with better technologies for the nanopores themselves. The amount of software that's available for nanopore sequence analysis is enormous, it's huge. Okay, so the role of whole genome sequencing and even metagenome sequencing in clinical microbiology will definitely increase in the coming years. So this will definitely transform clinical microbiology into a genome-powered and personalized diagnostic era. So with that, I thank you for listening.
Thank
That's
you.
cute.
Can you hear that? You hear that? Yeah. All right.
Yeah, I heard that. All 124 people cheering me on.
Thank you very much, Prof. For your
You're welcome.
and insightful presentation. So now I open the floor for questions. There's no questions in the chat box. I think they understand you very well, Prof.
Either that or they all fall asleep.
Wait, let me check if there's any questions. Yeah, thank you. Interesting sharing. Someone mentioned, HM mentioned.
Okay. Anyone doing nanopore sequencing? I'm really curious to hook up with anyone who has experience or doing nanopore sequencing, because I intend to start that next year.
So you do have the nanopore?
No,
Or
I don't. I just intend to, depending on 有钱 or 没有钱.
But Prof, your project you mentioned you did a hybrid sequencing.
Yeah, that one is doing PacBio. I sent for commercial provider PacBio sequencing, you see.
But PacBio considered as third generation as
It's
well.
considered as third generation, but it's really expensive. Like I said, Illumina costs about 600 ringgit per microbial sample. PacBio cost me 10 times, 6,000 plus ringgit. Yes. So that's why I really want to go into nanopore sequencer. I really want to hook up with anyone around here who's attending this talk, who has done or played with nanopore sequencing.
Yeah, HM mentioned that they only use Illumina.
Only used Illumina before, yeah.
I mean, my project is Illumina as well, MySeq.
MySeq. Do you guys have that in Utah, or you just have to send it?
We have a collaborator from Dr. Chan's lab, HIR, during the time.
Uh-huh.
Yeah, not as rich like the paper Smith, like he sequenced 400
over. Yes.
Gosh. Well, we only managed to do like 8 samples. It's not that easy to get mycobacterial tuberculosis positive samples anyway.
Correct. Yes, it's very, very difficult.
That's the limitation as well.
Yes.
And Prof, in your slide 34, you mentioned the estimated cost per sample.
This one?
Yeah. So what do you mean by flow cell that we use? So you can do that with cheaper price?
Yes. Okay, let me show the Minayon thing. Hang on.
Audiences, ladies and gentlemen, if you have any question, you may unmute your mic and ask the questions, or you may post your questions at the chat box.
Okay, if you look at this, this is the Minayon thing, right? So this is the flow cell. This is where the sequencing reactions take place. So when the Minayon first came out in 2006, 15, 16, I don't know when the Despicable Me came out. It's about the time.
I think they concept from there.
Yes.
Or they collaborate.
So the flow cell, right? You couldn't reuse them. So every time you buy them they ship from UK. You use it once, and after that you have to ship it back to UK. You have to send it back for them to recycle. Okay. So when the Minayon first came out, you can only use it only once, and they track every flow cell that they ship out. They'll track. And once you're done, because Oxford Nanopore works this way. I mean, I haven't done it myself, but this is what I read. It's like, okay, you see this? You hook it up to the laptop, right? Then you run it overnight, right? And then you let the reactions run overnight. And what happens here is that the raw data is actually transferred to the cloud. Right? So then you have what the program. There are programs called base callers that will convert those raw images into bases. GADC, 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 GADC. So all this is done in the cloud. Okay, so these Minayons, these flow cells, was single use. But I think starting from 2018 onwards, you have an option to reuse the flow cell, meaning after each run, you can actually wash the flow cell to reuse it. What I'm not sure is how many times you can reuse. That part I'm fuzzy about it because I haven't gone through the details about it yet. But you can reuse the flow cell starting from 2018 onwards. Okay, so that's what it's meant by when you talk about the cost thing. Yeah. Okay. So if you don't reuse a flow cell, it costs about $63, right? Flow cell reuse means you're recycling the flow cell yourself in your lab. So, but of course, it costs some, right? But definitely it's cheaper than the MySeq.
Oh, I mean, like as microbiologists or molecular biologists, it's quite deep. You know, the word like reuse your
Yes. Yes.
for any experiment.
Yes.
Yeah.
That's
Okay.
right.
Okay.
Okay. I see a question there. I can see that.
We have Dr. Wong Hanming, I think. Since your Acinetobacter baumannii plasmids are carrying mock genes, then do you plan to use a helper plasmid like PRK2013 to repeat your conjugation experiment?
Currently, no. Currently, no, because, well, now our lab is undergoing some, it's currently, what do you call it, out of bounds to more students, because before that we had the CMCO, all these kind of things. So currently, I think our wet lab experiments are basically put on hold for now. So that's why all my students are just basically doing bioinformatics work. Remember, I have 50 genomes to go through. What I did, the one I showed you just two. I did for big bio. Yeah. So that's a problem, you see. That's a problem about whole genome sequencing. You have a lot of data, and you just need people and time to go through and properly analyze them.
So in these questions, Dr. Wong, I mean, the helper plasmid will be able to change, I mean, to improve the results of the conjugation experiment?
Basically, I think because PRK 2013 is from what I know of PRK 2013, is basically is a helper plasmid because it has conjugative genes inside. So any other plasmid that has mobilizable gene will need a conjugative genes to help it mobilize to another strain, right? So in this essence, it's like it also boils down to the type of conjugation genes that can be mobilized by those plasmids. So as I said, it's a bit what do you call it? It's a bit complicated as well when you talk about conjugation mobilization. At this point, as I said, we tried doing direct conjugation, and that didn't work out. We tried many. We tried liquid media, we tried solid media, we tried different ratios of donor and recipient. It just didn't work. Not sure why. So, like I said, thank you for your suggestion of using PRK 2013. It's something we were thinking of using a different types of helper plasmids, but as I said, at this point in time, probably not likely for now. Maybe in the future. So yeah. So I see another question there up there. It says from where? From EC.
Yeah, from EC. Is Minion available in Malaysia so far? Are there any servicing laboratories for the technology?
Not that I know of, because Minion usually don't have servicing laboratories. Minion means because they do send you as a kit for the library prep, and then that USB drive. It's like a USB drive, the flow cell. So you do it yourself. So I mean, if you have you can essentially even do it at home because small paper. If you have
Or like you can carry anywhere and
you
you're
can carry it anywhere. It's portable. It's really portable. So if you have a set of Gilson pipettes at home and yellow tips, sterile yellow tips, you can do it at home, no problem.
you can start thinking of doing that.
Yeah, I can start thinking of doing that. That's why I want to do that. So because now you have to extract DNA and then you have to send to somewhere, I don't know, Korea, China. We tried so many service providers. BGI is not bad if they want to send to China, but it took so long. You only get back your raw data like what, more than 3 months after you send. So with the Menayon, you can get the data straight away. Uh, the price of a Minayon, that one is US dollars because Oxford Nanopore is based in the UK, so they quote in British pounds. So if you go to the website, you can actually get a price quote. I can't remember offhand how much it costs, but a starter kit, meaning that the library prep kit as well as the flow cell would I would estimate the cost about five thousand thereabouts. So, but the thing is, the thing about Minayon is they are sent directly from the UK to the country. I don't think they have what do you call it, a sales representative or something like that in Malaysia. So that one you have to deal directly with Oxford Nanopore
You are planning to start the
next
first.
year.
So how's the requirement for sample processing for Minayon? Like,
Minayon, of course, the biggest hurdle for us is you need to have really good genomic DNA quality. If you want to get good sequence quality, good reads, you need high quality DNA. I can't remember offhand exactly how much DNA or what's the quality, but I would assume that if it's good enough for Illumina, it should be good enough for Minayon. That's my assumption. I don't know. Maybe next year when you ask me to give a talk, I can give you more. I can give a what do you call it? A first-hand view of using the Menayon for whole genome sequencing.
okay.
Yes.
Great. I hope the audiences who are interested to look into working in Minayon, please do contact Prof. Yo.
Yes, you have my. I think my email. Where's my email? There you go. Yeah, you have my email on the screen there. Please, I'll be happy to hear from any of you who wants to do, not even Minaya, but any whole genome sequencing. Be happy to talk and discuss about that. Yeah,
Okay.
collaborations are always welcome, especially in these times where money is very, 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 very hard to come by.
Yeah.
Hey, Sihu, just curious. I mean, for the latest FRGS, how many grants that Utah obtained?
The latest, like,
The latest FRGS, how many grants did Utah obtain?
I have no idea.
No idea.
One from the floor.
Mhm. Anyone from the floor? No. I mean, how bad is it this year for Utah?
So it should be application this year, you mean 2019? I'm not sure about.
No, 2020 lah, because I think they recently announced only in when was it? Just November, wasn't it? They announced the results.
I have no data
No
about.
idea. No idea. Okay.
Sorry, Prof.
Okay.
Anyone here?
Hi, Prof. Utah can fund postdocs. Oh, I'm looking for one too.
Your requirement for students, is it like bioinformatics background or
My students all come from zero bioinformatics background. I forced them to learn Linux. I forced them to do per command lines, and they pick it up quite fast. They pick it up quite fast. I mean, it is not difficult. You're not doing coding. You're just using it.
That's the issue for scientists.
Yeah, exactly. Because yeah.
Okay, we have Dr.
Okay.
Hi.
Mega. Yes, I do use Mega as well. Mega is very useful.
Yeah. I like that.
For like for gene families and all that, I use Mega. But if you're doing like whole genome comparisons, Mega would can't process it. Definitely not.
Yeah,
Yeah, Mega can process it.
a free version of everything.
Yeah. Okay, I have yeah,
Brandon,
I'm you.
student. Hi, Brandon.
Hi, Brandon.
Slide 17. ST195.
195 went through clonal population shift. You're not sure why. That's a really good question, actually. We're really trying to figure out why this shift happened. My guess is my guess, like, because but this is just anecdotal evidence, because what we heard is that in 2011, 2012, there were like overuse of antibiotics in the hospital. So because if you look at ST195 isolates, some of them were actually resistant to even the polymyxins. They were all multi-drug resistant. And my guess is that the overuse of antibiotics may have sort of imposed a selective pressure for those population of 195 isolates to be maintained in the hospital during that time. Because when we look at the antibiotic resistant profiles, what we are seeing, although in most countries you're reporting increasing, but for the Terengganu hospital, strangely, especially for the past 3 years, we are getting a trend of decreasing resistance, not only for Acinetobacter, also for Staph aureus. So we are seeing a trend of decreasing resistance. And the hospital has actually implemented a pretty strict uh, policy of antibiotic use. So my guess is that actually helped to select for strains that are less resistant. So when you look at the population of Acinetobacter baumannii, say from 2015, 16, 17, they are all from various S sequence types. They're all from various clonal groups. And those are, most of them are not as resistant as the strains were back in 2011, 2012. So that's my guess. And because maybe the ST195 isolates, they may work well when there are other antibiotic resistant pressure, but maybe they don't have that what they call it, predominance, especially when resistant pressure is less. So that's my guess. But as I said, without empirical data, I can't say, because this is just anecdotal data. I don't have empirical data that yes, 2011, 2012, they're using this much of antibiotics in hospital. I don't have that.
Cool. Thanks, Brandon, for the questions.
Yeah, you're welcome. Any other questions?
Yeah, any other questions from the phone? So, Prof, how many samples you have collected so far in Terengganu, your total?
Total, we have about 400 plus.
Wow.
Yeah, no money to sequence all. My dream is to sequence everything.
Yeah,
I
wow.
have Acinito, I collect Acinetobacter from 2011, and then from 2015 onwards, I collect Staph aureus as well. But now I think Staph aureus is it's getting harder and harder to publish. I look for other type of staph. We have like the coagulase negative staph hemolyticus, staph hominis. So all these interesting staphs. We do get that, and looking at one that also have one of these large plasmids also. So the thing is now we don't have money for back bio.
Okay,
Yeah.
get some fun, Prof.
No, nowadays so difficult to find funds. Last year tried so many, all fail. Terrible.
Gosh, let's try again next year.
Yes.
All right, any
All
more?
right. Any more questions?
Really. Thanks, Prof, for
You're
your
welcome.
sharing. I hope, well, we can like open up or start some collaboration, yeah, with UNISA's,
Please,
perhaps.
that is very, very much welcome.
Perhaps that
Yes,
we may contact Prof. Yo, yeah, with his email.
yes, yes. Please do so.
Okay.
Okay.
Anything else? Audience, ladies and gentlemen, any more questions from you guys? No. Okay. So if there's no more questions, then let's end the talk for today, Prof. Thank you
Okay.
so much once again.
You're welcome. Very
Great.
happy.
This presentation finally,
Yes.
like,
Yes. After more than a year.
yeah. Thank
Although I'm supposed to be in Kampa, but can't help. Next time.
Next year. Let's
Next year. Next year. Next year. Next year. Let's hope for the best.
wish you all the best in your research, and thank you again.
Oh, thank you. You're most welcome. Thank you. Thank you.
All right, thank you very
Yes.
much to all for
Yes.
coming and your time. Thank you
Yeah.
so much.
Yes. Okay. Thanks, everybody.